Evaluation of parentals. Parental preparations are those preparations that are given by other than oral route. Essential requirements of parentals are they are needed to be sterile, free from pyrogens, free from foreign particles, and they are needed to be isotonic with the blood. The finished parental preparations are subjected to different kinds of tests like sterility test, leakage test, clarity test, pyrogen test, assay. And the first test is sterility test. Since all parentals are required to be free from microorganisms, they are to be tested for the sterility, isn't it? First principle involved in transferring the material under test aseptically into a nutrient media, and then it is incubated at optimal temperature and pH for a certain period of time. If it gives a growth of microbes, then that indicates the test is positive. If it doesn't give any growth of microbes, indicates the test is negative. If solid or oily materials are to be tested, they should be modified. That is, these materials produces some turbidity inside the media and does not enable one to be concluded that the turbidity is due to microbial growth or due to the material. So in such circumstances, what we will do means we need to further culture. So further subculture have been made by transferring a small quantities of a media under test into a fresh sterile media. If the preparation containing antimicrobial compounds like sulfatrax, penicillins, how we will conduct sterility test for such compounds means, so these are the compounds that itself have an antimicrobial property that doesn't allow microbes to grow. So how we will test such compounds means, simply we have to be tested under suitable antagonistic materials in direct amount into a media. Example, sulfatrax can be contracted by PABA, penicillins can be contracted by penicillinase. Some cases, samples taken for test can be suitable diluted to offset the antimicrobial effect. Example, phenol 0.5% can be mixed up with such an amount of media as dilute the phenol content by about 50 times, thus rendering ineffective in preventing the microbial growth. So like that we will conduct the sterility test. So furthermore checks the sterility of test media. So how means, by simply incubating the tube of a sample media and observe it shows growth or not. So or otherwise by inoculated within a chosen microbe and incubate and then check the growth. After transferring the material under test to the media, incubate it under specific conditions and then check the microbial growth whether it is sterile or not. However, the final decision in this matter should be taken straight two way, but differing under one or two tests are run successfully. So there always exists a problem of accidental contaminations from external sources during running of a test. So if sterility test fails a second time, a third time test should be run. If it fails for third time, then it is declared to be non-sterile with any degree of confidence. Next method is membrane filtration method. So it is accepted nowadays by USP for large volumes and small volume parenterals. It is subjected to a risk from environmental contamination. And method, the liquid to be tested is filtered through a sterile membrane filters of microbial retentive porosity under aseptic conditions. Then microbes are retained on the membrane filters while liquid being tested passed into the filter. Filter may be rinsed with a sterile liquid to remove the potentially inhibiting substances that adhere to the microbes. The filter is then placed in a culture media and incubate for growth. If growth occurs, it indicates that the product is non-sterile. If there is no growth, indicates that the product is sterile. Media, here we will use soybean casein digest media, thiocalculate media. Persons conducting the sterility test should be trained properly due to more complex manipulative steps involved in it. And the next test is pyrogen test. Pyrogens are the gram-negative endogenous toxins. These are the lipopolysaccharides which have a capability to induce fever. And the lipophilic lipid part induces fever and the polysaccharide lipid part induces solubility. They are thermostable, soluble in water, unaffected by bactericides, passes through bacteria-proof filters. So how we will find out these pyrogens? So the presence of the pyrogen substance in a parental preparation is determined by two types of tests. First one is in vivo, that is qualitative biological test based on a fever response of a rabbit. And the second one is in vitro lal test. Rabbit method, Here material to be tested is injected to the ear vein of the rabbits. So in this test we will pick up 3 rabbits and then inject the material to be injected, and then check the rectal temperatures of the 3 rabbits. The temperature should be 0.6 and should not be more than 1.4 for the 3 rabbits. After that we will conduct the test for another 5 rabbits, totally 8 rabbits. Here the temperature should be 0.6 or should not be more than 3.7 degrees centigrade. So like that we will conduct the rabbit test. And next test is LAL test. Lysites of amebicides of Limulus polyphemus. In presence of gram-negative endotoxin bacteria only, it forms a gel within 60 minutes when incubated at 37 degrees centigrade. So the drug present in formulation interferes with the fever responses. However, run the test on vehicles as well as a finished product also. Next test is clarity test. Contamination of a parental products with a solid particles or a fibers could be dangerous, especially if contaminants are larger than the blood vessels. Such particles causes a blockage in the blood vessels with consequent hazards. Clarity test is performed to ensure the parental products are free from foreign particles. The unlabeled containers are held by neck, strongly illuminating black and white screens. White screen is used for the dark colored particles. Black screen is used for the light colored particles. Visual inspection is done by inspection of the externally cleaned containers under good light viewed against white black backgrounds. Moving particle is easier to see than one that is stationary. Care must be taken to avoid the bubbles, which are difficult to distinguish from the dust particles. To see heavy particles, it is necessary to see by invert the container as first step in inspection. So although a human inspection is subject to decrease in efficiency by eye strain, fatigue, distractions, instrumental method of evaluation such as Coulter counters are used. In this instrumentation method, we will use the principle of light scattering, light absorption, electrical resistance to determine the particle count and the size distribution of the particles. USP establishes the limit of 50 particles of 10 micrometer and large per ml, 5 particles of 25 micrometers and large per ml, and the infusion should be free from 30 to 40 micrometer particles. Next test is leaker test. Containers of the parentra should be hermetically sealed, barring any communication between their content and the environment. This test is performed only for the ampules which have been sealed by fusion to ensure that there should not be any leakage in them. Tip sealing ampules are more likely to be incompletely sealed than the full seal ampules. It is performed in a vacuum chamber. Ampules are dipped in 1% solution of a methylene blue in a vacuum chamber and vacuum is applied. When vacuum is released, the color solution will enter into the ampule with defective sealing. The presence of the dye in the ampule confirms the leakage and hence rejected. Vials bottles are not subjected to such leaker test because of the fixability of the rubber closure and it is not resin. Next test is assay. It is performed according to the method given in the monograph of the parental preparations in the pharmacopoeias. It is used to check the quantity of a medicament present in the parenteral preparation. This is about the evaluation of the parenterals. Thank you for watching this video. Please subscribe to our channel for more videos.